Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rozan, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the question and answer portion, and I will ask our speaker your questions from the window. Your questions will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Eric Bishop from Cygnus Technologies.
Welcome, and thank you all for taking the time out of your day to participate in this webinar. Today we're going to discuss some of our work and services that incorporate mass spec into host cell protein analytics. More specifically, how to use mass spec to truly understand the HCP in your process and to make sure that you are using appropriate HCP ELISA for process monitoring. Oh, good. First, a brief history of Cygnus Technologies. We became involved in HCP analytics in 1998 with the publication of a white paper with our idea of how to monitor HCP. In 1999, we launched our first commercial CHO and E. coli host cell protein kits. We spent much of the early 2000s expanding our menu of generic kits to cover all of the commonly used expression platforms. And then after performing 2D Western blot for coverage analysis for a number of years, we realized that it was not predictive of how the antibodies would perform in the ELISA. As a result, we launched the Antibody Affinity extraction in 2014. This method removes many of the limitations associated with 2D Western blot. We spent a lot of time in the past few years working on the integration of AAE and mass spec into host cell protein analytics. I plan to spend the next few minutes describing this work and will hopefully illustrate the importance of truly understanding the HCPs that are in your process. A little bit about the actual host cell protein ELISA. On the left is a schematic of how an HCP ELISA works. This method is considered the gold standard for process monitoring and release testing for HCP, primarily because it's the one method that we have that can measure nanogram per mil levels of HCP in the presence of milligram per mil levels of product protein. It also does not require a special level of expertise, and it's easy to transfer across an organization. Unfortunately, the host cell protein ELISA also has some limitations. At the end of the assay, you'll read the plate and the software reports a result for each sample. Unfortunately, this result does not tell you anything about what HCPs are in the sample or which HCPs the assay reacted to. Also, while it does not require a high level of expertise to run, it does require a high level of expertise to develop a broadly reactive HCP antibody and then to turn that into an accurate and robust HCP ELISA. Neither of these points are lost on regulatory agents, and as a result, they want you to make them feel comfortable that if you're reporting low levels of HCP, it's because you have low levels of HCP and not because you're using an insensitive method. This requires the integration of orthogonal methods such as antibody affinity extraction and mass spectrometry. So next we'll spend just a minute discussing antibody affinity extraction. So as I mentioned in the intro, AAE was launched in 2014 as a method that was much more predictive of how antibodies would perform in an HCP ELISA than a 2D Western blot. This diagram is an illustration of how AAE works in a nutshell. Initially, the same antibodies used in the assay are coupled to a solid support. After extensive conditioning and washing, the sample is loaded. When performing AAE for coverage analysis, the sample is a null or a mock transfected harvest sample. After loading the column, it is washed until the background signal is achieved, and then it is eluted. So at the end of the experiment, there are two samples. The first sample is the starting material, and that represents all of the HCPs that could find their way into your product. The second sample is the AAE eluate. This is all of the HCPs that were specifically removed by the antibodies on the column. And for the past few years, the most common detection method has been large format 2D silver stain. However, more recently, many companies are opting for mass spec detection for coverage analysis to avoid many of the limitations associated with gels. Here is a representative data from an AAE fractionation and a 2D silver stain detection. The image on the left is the starting sample, and the image on the right is the AAE elution fraction. The first point I would like to make is that the sole reason to perform coverage analysis is to demonstrate that the antibody used in the assay is broadly reactive to the HCPs in the process samples. In this example, no real analysis is even required to say that this antibody is broadly reactive to the HCPs sample. Second, we ran this sample for 6 cycles over the AAE column to make a point. And that point is that AAE also acts to enrich HCPs, which is very important to the next part of this discussion. Finally, if you focus on the low molecular weight proteins, you'll see that not only did the antibody react to all the HCPs visible in the starting material, we were also able to detect many more proteins in the eluate sample. This is important because many companies think that in order to make antibodies to low molecular weight host cell proteins, that you must fractionate the proteins themselves. These images are evidence that using the proprietary methods employed by Cygnus, broadly reactive antibodies can be made to even the smallest and most non-immunogenic host cell proteins. So to close out AAE, AAE is a superior method to predict how the antibodies will perform in the host cell protein ELISA. It can enrich HCPs and deplete product proteins, so it can also be valuable in the analysis of downstream samples. And AAE is an excellent sample preparation tool for mass spec. So now we will examine AAE mass spec case studies, just to provide a flavor of what we're doing with mass spec and some of the potential problems that we can address. This slide illustrates our typical AAE mass spec workflow. It starts with AAE fractionation, then sample digestion, mass spec injection, and data analysis. So as I alluded to in the AAE discussion, mass spec is more and more being used for coverage analysis. This slide is a very simplified graph to demonstrate the coverage of the 3G CHO antibodies to the antigen in the kit is 90% by mass spec. That coverage number can range from 55 to 90% depending on the method employed, but that again is a discussion for another time. In this example, we have aligned the HCPs identified in the starting sample with the HCPs identified in the AAE elution fraction. For many years, companies have asked us if the HCP ELISA could detect some specific protein. And to be honest, the answer was always, maybe. We knew the antibody was very broadly reactive, so it would most likely detected the protein, but there really was no way to know for sure. So with AAE and mass spec, we can now definitively answer this question. So here's an example with PLBL2. The bar on the left is detection of PLBL2 prior to AAE. The bar on the right is PLBL2 in the post-AAE sample. And you can see that PLBL2 has been enriched by 20-fold, and with that information we can feel very comfortable that the assay is reactive to PLBL2. This is a slide that I believe eventually will change the entire industry. As an industry, we have always known that mass spec would be a great tool in understanding HCP. However, the bar graph on the left demonstrates why mass spec has had limited success in HCP analytics. As you can see, the product-related proteins are in such abundance that the detector is swamped and very few HCPs are detected. The image on the right is visibility when incorporating AAE as a sample preparation tool for mass spec. As you can see, the product-related peptides have been greatly depleted and the HCPs have been enriched. This means that for the first time, we truly have the ability to know what HCPs are persisting through the purification process and into the final drug substance. This knowledge will allow us to perform risk assessments on identified HCP and to make decisions about which HCPs have the ability to cause negative effects with respect to patient safety and product stability. This slide is a good demonstration of how you can use mass spec to supplement the data package used to justify the use of a particular HCP ELISA. The proteins on this table are known to be problematic proteins in one way or another. The blue bars are proteins prior to AAE. The red bars are post-AAE. And as you can see, that each of these were enriched, and therefore we can feel comfortable that the antibody used in this kit specifically detects each of these problematic proteins. And also one more point, as you can see, the 5 red bars on the end were not detected prior to mass or prior to AAE. So again, AAE just gives us an additional insight that we did not have before. Finally, AAE and mass spec can be very helpful in investigations of HCP problems. This is most often due to the enrichment of a single HCP by the purification process and presents as a loss of dilutional linearity. With mass spec, we can test samples that have good linearity and compare that with a sample where we have lost dilutional linearity and typically can identify the offending protein. The mass spec also provides information such as PI and molecular weight, which can be passed to the process development engineers, and the problematic protein can be targeted for removal. Companies often come to us because they understand that mass spec is important for HCP analytics, but they are unsure of when or how to do it. So we put this slide together to illustrate that it may be useful to consider when it may be useful to consider mass spec for host cell protein analysis. As you can see, mass spec can play an important role in HCP analytics from IND through post-marketing. This can include coverage analysis, detection of hitchhiker proteins, evaluating the impacts of a process change, the full risk assessment, and also characterizing reagent changes, and this is just to name a few. So to wrap up, incorporating mass spec into HCP analytics can be a highly valuable strategy to ensure the best possible clinical outcome. You can assure that the HCP ELISA is fit for purpose, as well as to enrich and identify hitchhiker protein. It is also a valuable tool to guide purification process development and to assess the impacts of process changes. However, the most important use for mass spec is the ability to identify the HCPs that are found in the final drug substance. AAE coupled to mass spec is the best tool available to date to accomplish this task. With this information, it is possible to fully understand the HCPs that are in the product and to have an idea of how that could impact patient safety and drug stability early in the project and to ensure, again, the best clinical outcomes. So I'd like to go ahead and stop here and then take any questions that you may have. Thank you.
Great, thanks, Eric. So can you perform antibody coverage analysis on the product containing harvest material?
So you can do coverage analysis if product is present, but you're limited in your detection method. So if you have product, you are limited to doing it by mass spec, because with AAE, when you have product present, it's still 90-some% of the total protein, and most host cell proteins are not visible on gel. So if you do mass spec analysis, then yes, you can.
Okay. And for AAE, how do you capture your antibodies, and how do you ensure that no antibody isotypes are preferred?
So, well, that's kind of getting into some of the proprietary parts of how we do AAE. So I'm not so sure that I can get into some details on that one, actually.
Okay. And do you do both intact protein MS and peptide mapping?
We do intact.
And how much sample milligram is required for AAE?
It depends on what we're doing. We try to not think of it in terms of the product because the product is not what we're looking for. So we need to make sure that the samples that we receive have at least 1 to 500 micrograms of host cell protein. So the amount of product varies depending on where you are in the purification process.
And do you recommend performing AEMS coverage on both upstream harvest material and final drug substance?
Absolutely. So those two samples are really important for different reasons. So for the upstream coverage analysis, it's important to show that the antibodies in the kit that you're using are broadly reactive to all or at least most of the host cell proteins that are potentially going to find their way into the product. And of course, doing it on the drug substance is the most important sample you can do because you can identify what host cell proteins are actually in your final drug substance. And again, with that information, perform a risk assessment and make decisions about if this is acceptable or if something needs to be removed.
And can you perform quantification of HCPs in the final drug substance using AAEMS?
We can. So if we want to quantify the host cell proteins, we have standard proteins that we can spike into a sample and then measure or quantify the host cell proteins, the unknown host cell proteins based off of those standards.
Okay. Can you please explain on slide 13, AAE is detected with less protein compared to the ELISA technique?
Can you say that again?
They say that AAE detected less protein compared to the ELISA technique. Can you please explain?
Oh, I'm not really sure what that question is asking. All of this is mass spec data, and it's relative to the most abundant protein. So again, the blue bars are each of these proteins detected by mass spec without doing AAE, and the red bars are doing protein identification post AAE.
Okay. And how do you deal with hitchhiker HCPs to drug substance?
Well, to be perfectly honest, 90% of the time, or probably 99% of the time, when we actually do additional investigation, we find that the host cell protein is not cross-reactive. We have tools at our disposal that we can, make that determination. If it is cross-reacting to the product, then obviously there's some additional work that needs to be done to resolve that. But most of the time, it's not truly cross-reactivity.
Okay. And I think we have time for just one more question. If you have additional questions, you can go ahead and enter them now in the Q&A, and we'll pass those on to Eric, and he can follow up with you directly. Or, of course, you can direct them to the email address on your screen. So the last question for now is, what's the sensitivity of AAE in terms of nanograms of HCP?
With respect to individual host cell proteins, I assume, to be perfectly honest, I would have to talk to my mass spec scientist. And that's the other thing, when you direct your questions to us, it's going to actually go to the mass spec scientist that can get into the more technical details of the method.
Okay, great. Well, thank you, Eric.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.